So I've been, for the last few years, trying to find ways to integrate what we know about structural biology with their different applications of systems biology, and I, by and large, have been unsuccessful in the sense that I think most people working in systems biology know as much about structure as I know about statistics. But I'm hoping over time, as communities emerge, then that situation changes. At least I'm learning some statistics. So what we'd like to do is to put a structural face on protein interactions, protein networks, et cetera. And what I'll do today is first introduce a program called PREPI that we developed a number of years ago. And I'll show you how we get there and what we do, because I want to give you a sense of how we think about problems and how we use structural information. And then I'll rapidly go through a number of applications, hopefully to interest you in what's becoming increasingly possible with the use of structure. This is a slide I've shown for many years, and it just makes a point of how structural information might be used. These are 3 different proteins, 3 different functions, and they all look differently. But if you look at substructures within the protein, geometrically, they're very similar. And it turns out that in each of those proteins, within those substructures, there's a metal binding site, all cations, different cations. But when looking at this, might suspect that, well, either it was divergent or convergent evolution. One way or another, there's information in looking at substructures in protein to try to get function. We talk about this as using structural blast in the sense that instead of looking for sequence motifs within structure, we're looking at structural motifs that may be small ones. And that's basically how I'll be using structure in this talk, looking for common structural motifs. Now, getting to apply this sort of logic on a genome-wide scale, there are a few issues that need to be dealt with. The first is there are on the order of 5,000 structures of human proteins, not complete structures, but at least of one domain in a protein, in the protein data bank, the repository of protein structures. But if one builds what are called homology models, where you take a sequence of a protein of unknown structure and map it on a known structure, you can build on the order of, say, 13,000 models. When you add up these 2 numbers, you get a decent structural coverage of the human genome. So we use homology modeling to build up our repertoire of structures, and that's point 1. The way we apply that information to protein, prediction of protein structures is summarized in this slide. We can start with 2 sequences of 2 proteins, and we ask, do they form a complex or not? Now, in this case, we know a structure of one of the domains of this protein from the protein data bank, and we know the structure of another from a model. Now, we have a structure in the protein data bank of a complex of different proteins. We now geometrically superimpose the proteins that what we call our query proteins on the known complex, and we get what we call an interaction model a predicted model of how these 2 proteins might interact. And a crucial element is this geometric superposition. There are many mathematical algorithms that superimpose structures. It's a pretty well done field. So now we have what we call an interaction model, and we have to decide whether we think it's a good model or not. And that becomes the second issue we have to deal with. There are all kinds of physics-based methods to evaluate a model. What's the interaction energy? There are all kinds of calculations you can do. But that takes a lot of time if you're looking at hundreds of millions of possible interactions. So we have to come up with a faster method. But let me give you an overview of the pipeline that we have, and some of this repeats what was in the last slide. We start with 2 sequences We ask, is there a model, either a structure or a homology model for those proteins? And now we look through the protein data bank for related structures, and we do that again by superimposing structures. So we're looking only for geometric similarity, and we find an average about 1,000 what we call neighbors for each of these, which means on the order of 1 million pairs of interaction. If any of these pairs are in the protein data bank, we have a model of a complex. We then superimpose our unknown structures, or our query structures, onto that complex and score it in a way I'll go through very quickly. PrEPI also uses non-structural evidence, I'll see in a second, sequence relationships, functional relationships, co-expression, etc., and combines all this information in a Bayesian framework. And that's very important in the sense that we're using structure, we're introducing it, we're using machine learning to evaluate structures, which is something that really hasn't been done before. The way we score our models, I'm just giving you a very crude sense of it. Here's two proteins who form a complex that we know. We know what amino acids are in the interface. We superimpose our structures on these structures. We ask, how similar are the structures? How similar is the yellow to the green? And there's a measure of that. We ask, how many common amino acids in the sequence are there in the interface? And we ask if the amino acids in the interface are amino acids that statistically are likely to be in interfaces. Certain amino acids, such as tryptophan, are very often in interfaces. Lysines are not. So we have a statistical measure which we get a score from, what we call a structural modeling score. And then we, using positive and negative data sets, we compute a likelihood ratio that this complex between the light blue and the light green actually occurs. As I said a few minutes ago, we combine structural scores with other sources of information. I'm not going to go through them all, but excuse me. Oops. What am I doing? Well, I'm going through the talk quickly, but in the wrong direction. I have thick fingers. I didn't know they were that thick. Yes, so these are the various, oh, Jesus. I apologize. Various sources of information. Each one of them is trained on a positive and negative data set. And when we're through, the new version of PrEPI. PrEPI stands for Predicting Protein-Protein Interactions. Some of you who were both born in this country and are as old as I am will know that PrEPI has another meeting, but most of you won't, so it's supposed to be funny. Anyway, we have in the PrEPI database, here I'm doing it again, about over 1 million predicted interactions. Those that have a likelihood ratio of above 600, we know from our experience and from other sources that that's what we can call a high confidence interaction. In the PrEPI database, there are about 1 million interactions that don't appear in any other database. And of these, we believe that about 500,000 may be direct physical interactions, as based on having a PrEPI score of 600 and a structural score of greater than 100. These numbers won't mean much to you, but I'm just sort of telling you that we have measures for this. To give you a sense of, this is the PrEPI database, which can be queried. You can find the sources of information used for the prediction. You can look at models. SNPs or cancer mutations are mapped to these models. So there's a great deal of information in the database. This is a rock curve, which we've used PrEPI on a database compiled by Mark Vidal's group. And what you can see here at the low false positive region is that our predicted interactions are as accurate in all the tests that we've done as a yeast-2 hybrid experiment or mass spec, or data taken from a mass spec database. But we have over 1 million of such interactions, and all you have to do is click to find them. This is on the same database. This is a database that Vidal's group compiled of known interactions, and this is the performance of PrEPI as a function of LR. And you can see that at an LR of 600, which we use, we are getting actually lots of good predictions. And for a negative data set, the RRS data set, at 600, we're not making many mistakes. And these lines show you what the mass spec Bioplex and yeast-2 hybrid database, both from Harvard, what coverage they get. So we think we have a unique resource, and we want to use it in different applications. When we first published the PrEPI paper, much of our validation was statistical, but we did some experiments with collaborators, and just remembering 5 years ago, we made 19 predictions, 15 were validated with co-IP. In unpublished work, we're collaborating with Sagi Shapira and Gorka Lasso, a postdoc who Sagi and I share. And these are predictions of interactions between viral and host proteins. So you probably can't read this. I can't. This is the virus. Is the virus protein, human protein, and LRs. And the point to see here is that above an LR of 100, which we would call a positive prediction, everything in green was validated. Everything in orange was a false prediction. And if you go below 100, you can see the success rate is obviously down. But this is unpublished validation, not on human proteins alone, which we had been studying, but on viral host interactions. So we have confidence that many of our predictions are correct. Now getting on to the evaluation of SNPs, or different mutations, we looked at the ClinVar database, and we asked, are proteins that come from the same disease likely to interact with each other? And out of 3,700 proteins from the same disease, we predict that 800 physically interact with each other. If they come from a different disease, you can see the numbers way down. And we just took random sets of proteins. It is also way down. So this is an important result. We're not the first people to make this observation, but we're making it based on predicted interactions that proteins from the same disease, maybe not surprisingly, are predicted to interact. We then looked, and we asked, are disease mutations do they preferentially appear in PrEPI predicted interfaces? And you can see there's an enrichment of mutations in PrEPI predicted interfaces. And for benign or SNPs taken from the 1,000 Genomes Project, there's a decrease in the number of interactions predicted by PrEPI. So PrEPI seems to be finding relationships between disease mutations. Now we're using that, and this is really very preliminary, in the following way. We'd like to be able to predict whether a particular mutation, say, is associated with cancer or not, and in particular look at rare mutations. So what we've done is construct a network of proteins, let's say we're interested in a particular protein P. We have a PrEPI network. We have mutations, and we ask, do different mutations disrupt interfaces either of the protein we're interested in, of proteins that are neighbors of that protein, or proteins that have the same function shown here in yellow. We just count the number of interactions that are disrupted, presumably, by a mutation. We do the same thing for benign mutations. We taken from the 1,000 Genomes Project. We put this in a random forest program, and at least for this one case here of which is from breast cancer, are highest, our best predicted mutations, there's a great enrichment in breast cancer patients versus sort of random mutations from people who don't have the disease. So this is not published, it's preliminary. I'm just trying to give you a sense of where we're going, and using structural information, we're able to ask functionally what is the role of a mutation and put that into an algorithm to make predictions that you couldn't make if you didn't have a structural context to interpret your results. Now, we've begun to, Andrea Califano was a collaborator in the original PrEPI paper, and we continue our collaboration very closely. And we've come up together with a student, Josh Broyd, and a collaborator, Diana Murray, to try to incorporate PrEPI networks with Andrea's regulatory networks, about which you heard yesterday in his talk. So here is just a hypothetical PrEPI network centered on a particular protein. And what we do then is take that PrEPI network and combine it with Viper, Cindy, and Arachne, also centered on the same protein. We take all these predicted interactions. Let's say this central protein is KRAS. We have a gold standard set taken from known KRAS pathways, and we train our system. I mean, we get predictions from the random forest algorithm. And when we're through, what we have here is what we call a protein-centric signaling map, in this case for KRAS in lung adenocarcinoma. So PrEPI is giving us a canonical protein-protein interaction network, whereas the various regulatory networks coming from Andrea are tissue-specific, tumor-specific, et cetera. So we've combined physical information with regulatory information. We come up with, in this case, KRAS-specific pathways, which certainly add directionality from our perspective to PrEPI. These are upstream, these are downstream, and you can see in some cases there are what look like feedback loops. Of the 40 top hits, I'm not showing you the details, that we make, 18 were already known, but of course they weren't, they're not part of the information used to derive them. And of the novel predictions, 19 out of 22 were validated by synthetic lethal in experiments of Alejandro Sweet Cordero at Stanford. So this is just, again, a taste of what we're able to do, which we think is fundamentally new, having tumor-specific, cell-specific pathways with physical interaction included. Now, this is a blow-up. The last slide didn't provide any information. This is a blow-up of what we'd call OncoSig, an OncoSig network. And what I want to point to here, if I can find it, is this upstream modulator of KRAS, and then KRAS then makes direct physical interaction with this protein NCF2L. So we have a loop here, and I've simply highlighted the proteins in that sort of loop or pathway. And we now want to use PrEPI and want to use the structural information we have to try to get some mechanistic insight about what's actually contained and what's going on within this pathway. So this is a slide which gives you a sense of what's going on. The upstream protein, I don't know, DERK1B, has a kinase domain, a serine kinase domain. PrEPI predicts that it interacts with a kinase domain in trio, and we're able to build a model of that interaction. Now, it's interesting. These are multi-domain proteins. So the next interaction, of trio with MCF is not through the kinase domain, but through a Rho-Gef domain interacting with a pH domain in MCF. And finally, a different Rho-Gef domain in MCF is interacting with KRAS. So we actually can take ourselves, in this case, through the domains and ask, and get a mechanistic picture of why this loop emerges. Interestingly enough, oops, go again. Some of these interfaces look like they might be druggable, some may not, but in principle, we have targets for drug discovery efforts based on the structures predicted by PrEPI, extracted from this network. Now, in a moment, in the last part of my talk, I'll be describing how we use the same concepts I've been talking about to predict what drugs might bind to a particular protein. But here I'll just point out that, in a way, the result of what I'm about to show you is that a known drug, you can submit is predicted to bind to DYRK1B. Actually, not only is it predicted, but it turns out that this was known. And we have possible predictions for what proteins bind to the serine 3 kinase of trio. So, but to go from structure to drugs is something I'll finish my presentation on. This slide makes a point that some of the other interactions don't have binding sites that are obviously druggable. What's been done here is to superimpose the Rho-Gef domain of trio with the Rho-Gef domain of MCF. You can see this structural superposition of the two domains. PrEPI predicts that one of them will interact with KRAS here, and another of the domains interacts with the pH domain of MCF in a different location. So if one could successfully drug protein-protein interactions, which I'm sure many of you know has been a difficult challenge, that of course gives us a totally different way of going at pathways and trying to disrupt pathways. So how do we relate structure to small molecules, and how do we use these tools? So this is an example of a starting program, really. Let's say we have a protein shown here, and we'd like to know where it binds a small molecule. I'm not asking yet what small molecule it binds to, just where it binds a small molecule. So what we do is we have a database of other proteins bound to small molecules in ways that we know, and we structurally superimpose our query green protein on these orange proteins. And that superposition actually places each of these small molecules on the surface of our query green protein, shown here. And then we can simply look at the numbers of interactions made, the amino acids that are in the predicted interfaces, gather statistics, and say, for example, that this is a hot spot here for protein small molecule interactions. We do a little more than that. We actually ask, I'm not going to go through this slide, we ask, is the binding site of our unknown protein compatible with the, similar to the binding site of the protein that we know is in a complex. And when we do that, we can, as a precision recall curve of our program, L-bias, and of course, in our hands, all other protein programs are inferior. This is a well-known problem, but we hope it's meaningful. And you can see that we do very well in predicting the location of binding sites. Now, predicting binding sites is just part of the story. what you'd really like to be able to do, well, there are a few things you'd like to be able to do. So the second problem we'd like to solve is to ask, given a known drug-protein complex shown here, let's say this is a real drug with a real protein, people are interested in off-targets, what other proteins might it bind to. So the standard way of sort of approaching this problem, I say the most common way, is through what's called docking. You might take this drug and try to dock it onto the surface of every protein in whose structure you know. And that method works at times, it doesn't work at times, in part because the structures are of uncertain quality unless they come from the protein data bank, in part because there can be conformational changes on the surface of the protein as a result of drug binding. So in the spirit of what I call a lower resolution approach, which can generate hypotheses, we do the following, very similar to what I was just talking about. We superimpose the green protein. Now we know what this protein, we have this structure, we superimpose it on every protein whose structure we have in the human genome, and we get a crude model for how our drug will bind to every other protein. We calculate a score based on the nature of the binding site, and we come up with a prediction. Whether this is good or bad performance is it's actually quite good if we compare it to others, but I want to make a point here. We think this is a good start, that if you could do the same thing with sequence. You could take a protein with a known in a complex and ask what other proteins in the genome have a similar sequence to that protein, and say, are they possible targets for the same small molecule? And this is what I shouldn't be allowed near anything technical. This is what the predictions you get from sequence alone. This is what you get from structure alone, and the orange is just combining sequence and structure. So this gives you a sense of how much more information comes from structure relative to sequence. And for those of you that aren't structural biologists, that means most of you or all of you, except for the people from my lab, it's not a surprise to anyone who works with structure, because we know that structure is much better conserved in sequence, and there are many structural relationships that simply you'll never find by running BLAST. And this is the advantage we're getting from, or one of the advantages we're getting from using sequence. So now, finally, there's another, here is an example of a drug that's known to bind to KIT, and here's just a prediction of another protein. It binds to LRK2, which we predicted. Then, of course, we looked in the literature and found out that this was known. But this is an example of known structure, model, prediction. And the prediction, not the nature of the interaction, but the existence of the interaction was validated, in the literature. Finally, what I've talked about so far, and I'm at the end of my talk, is navigating through structure space based on structural similarities between proteins. If these proteins have similar structures, then it's possible, and even likely in many cases, that they'll have similar properties. So these are sort of protein-protein, if you will, similarity networks. But let's say there's another problem we're interested in. Instead of starting with a complex and finding out what are the off-targets of the small molecule in that complex, we can ask a different question. If someone does a phenotypic screen with a small molecule, what are the targets of the small molecule? So that's a different question. And to do that, one would want to navigate not through structure space at the beginning, but through chemical space. So here again are different compounds that they're all shown the same, that are similar as chemical entities. And there are various ways that people, primarily in the pharmaceutical industry, have developed to measure chemical similarities. So we could start with, say, if this is the small molecule you're interested in, these are other similar small molecules. You can make that relationship. But to make a connection to protein space, you need a complex. So the notion is that we could start with a small molecule, find similar small molecules until hopefully we find one that binds to a particular protein, and then navigate through protein space to see other possible targets of this small molecule. And this is what we've now implemented. I don't have results to show you, but ultimately the notion is to go from compound in red to protein in red, or from protein in red to compound in red. So by being able to navigate through structure and chemistry space, this application is going to be increasingly possible, and we're hoping over the next few years to be able to integrate these tools in various research programs, both at Columbia and at other places where finding targets, relating targets to chemistry will become increasingly valuable. So that's it. I'd like to, that's it. Mention the people that have been involved in this work. Donald Petrie is a senior scientist who's been in my lab longer than either one of us likes to remember. Cliff Zhang was a graduate student who wrote the first version of PrEPI. Nacho Garzón wrote the second. Diana Murray is an independent scientist, but she collaborates with us and with Andrea extensively, and is also responsible for our CAS outreach program. Josh Broida was a shared graduate student in mine and Andrea's lab who developed the ANCA-sig algorithm. Gorka Lasso has done the calculations and adapted PrEPI to host virus host interactions. Hawuk Wang is a senior postdoc who's really done all the work on chemistry that I've mentioned. Andrea, I've mentioned, interacted PrEPI and ANCA-sig, and Sagi Shapira on the study of virus host interactions, and thank you for your attention. It's Italian, isn't that what it's
I'm from Finland.
Great talk. I have two questions. First is, how do you see we can extend this prediction framework to include a higher order interaction information, like additive binding or forming a protein complex? Second is how to include the post-translational modifications into the picture.
So the first question is a direct pathway to that, because once we have models, I showed you one example where ROGEF binds to KRAS and to MCF in different locations. In many other cases, they can bind to the same location. In fact, we're working on a project to use, introduces, not that it introduces, but talks about the concept of cancer switches of competition based on structure of the same binding site. Post-translational modifications, I hate when people ask me that question. We're aware of them. We're sure if we know where phosphorylation sites are, we can. So, well, let me answer the question in a very different way. Everything I've talked about, the focus has been genome-wide. So we're using structural information in very different ways than it's traditionally used, because traditionally, crystallographers, for example, care about every kilocalorie and every angstrom. And we're just building cruder models, but we're sort of in the business, if you will, of hypothesis generation, the work I described. If the hypothesis is interesting, then you have to look at the biology, you have to read the literature, you have to add that information and refine your prediction. These are starting points. If the prediction is interesting, then hopefully it'll be worth the effort to refine it and go to the next level, including post-translational modifications.
So when you look at these binding interfaces in cancer, are they somatically, is there any selection pressure, both positive or negative, in terms of, you imagine like there's a, keep one and then FE2L2 is, for example, protein-protein interactions that's disrupted by recurrent mutations in lung cancer. With your predictions, you could probably approach that much more generally.
I think that's correct. I admit we haven't gotten there yet, but that's a very interesting application. So I'll just say thank you.
How is it to incorporate specific mutations, for example, in the disruption of these interactions?
So right now in what we've done, we assume that every mutation is disrupted. The next level is to say, is it a conservative mutation? Is it not one? We haven't done that yet, but that's again down the path.
But it could be the other way around. It can be that it's even activating or just
It could be, and that's where, if one non-polar amino acid makes a bigger one, well, it's hard. You see, you have to look. If you say have a mutation with a larger amino acid, it could make interactions stronger, or it could sterically get in the way. That's where you need to look at each one with some higher level of refinement.
So the other question was, when you were looking at the virus-host interaction, so there were some players, like STAT3 appears interacting with several. Can you comment on this? I mean, do you think they're sticky proteins, or they're specific because they're important for the virus? Or, do you think it's functional, or is it just something because it's structural?
I saw the slide last night.
Oh, I see.
But when we work on the paper, we'll be able to address that question.
Yeah.
So, you listed that there's at the beginning you have predictions like 20,000 human proteins, but one of the favorite things to do in RNA is to go looking for alternatively spliced transcripts, some fraction of which are presumably protein coding. And so when you start incorporating all the alternative starts, alternative stops, skipped exons, whatever, you're looking at a much, much bigger space. How do you account for that at all? Is that possible to do? Is it just
One day, but it's really beyond the scope of what we're doing now. We are using crystal structures to start. So whatever has been crystallized is our starting point, and then the models that we build are on domains or sequences that can be superimposed on those structures. I'm obviously aware of all that, and the other part of my lab, I worry about all of those things in a particular project, but it wouldn't be feasible at this stage of development.
So, question there. So,
Califano.
Okay, Califano.
I shouldn't be asking a question because I'll probably ask you, afterwards. But so it's really exciting the quadruplex, the interaction that you've shown there. We actually also highlight some potential, autoregulatory interactions that may exist. But how difficult is to extend that type of analysis to be done systematically across the entire proteome? Because I think some of the things that we had discussed in the past was that actually finding complexes from the interaction of pairwise proteins was not a simple thing. And I was wondering how much manual labor is involved in identifying, highlighting those kind of interactions.
I think we can do it, at least just starting with this information, going back to the domain structure of proteins. I think we can do just that.
That's good.
This is really a new thing that Diana came up with, and we're going to discuss with you on Thursday.
Because the problem is that everybody knows the complexes that are in some sort of stoichiometric equilibrium, right? Because those are easy to identify. But the ones that are both either transient or involved in single transaction and are relatively small, so they don't necessarily correspond to gene expression, those are much harder. I mean, if the method could be extended, it would be, I think,
we can do
major.
that.
Yes.
From the post-translational modification, are you aware of the oligomerization I mean structure? Because sometimes, cancer models, mutation or phosphorylations can lead to a different oligomerization of the protein, and that may be causes translation cell complex formation that could lead to a response that it's very critical for the self-regulation.
But Marie, was that a comment or a question?
And the comment,
I agree with your comment
yes.
completely, yes.
Because I know, well, structural biologists, most of Korean people, they're doing that. So I was wondering if you're aware of that in your model.
Looking at
Yeah,
oligomerization,
oligomerization, yes.
the experimental part of what I do is actually looks at oligomerization of proteins and cell surfaces to predict oligomerization based on post-translational modification is something that, to my knowledge, hasn't happened yet. One step at a time.
It's just curiosity.
No, it's a fair question. I wish I had an answer.
Well, thank you.